Hi, this is Dr. Kat Vlies from Central New Mexico Community College. We're going to focus in this video on the neuromuscular junction. In the previous video, I explained to you that a neuromuscular junction is a synapsis between an axonal terminal with its synaptic bulb of a somatic motor neuron and a skeletal muscle cell or a muscle fiber. And so here I'm drawing the sarcolemma, and notice that nearby that axonal terminal, it is very convoluted. This right here illustrates a T-tubule, and perhaps there's another T-tubule over here. That also means that I need to add some of our sarcoplasmic reticulum, and rather than drawing all of the sarcoplasmic reticulum, I'm just going to show you the terminal cisternae as these sac-like structures like so that give rise to this tubular system. And on either side of these T-tubules, remember, we always have a terminal cisterna. TC for terminal cisterna, T-tubule right here, okay. Here's our axonal terminal, and inside of these axonal terminals, or synaptic bulbs of these axonal terminals, we have little vesicles, and we call these synaptic vesicles. They contain neurotransmitters. And the neurotransmitter that is always going to be released onto skeletal muscle is going to be acetylcholine, always. It will never be any other neurotransmitter. There are many neurotransmitters in our body, especially in our central nervous system, but pretty much throughout our body. And some examples that you're familiar with are norepinephrine, dopamine, serotonin, just to name a few. But the one that always lands on skeletal muscles and that is always being released by the synaptic vesicles of our somatic motor neuron is acetylcholine. So or I should say, if this is our synapses right here, then right here, this cleft, this little bit of a space, we express it in nanometers, so it's very minute. We refer to that as a synaptic. That's the synaptic cleft. And it is just filled with the material that you find around any cell, namely extracellular material. Extracellular material in the synaptic cleft around our cells, and therefore also inside of the T-tubules. So your T-tubules are filled with extracellular material. It's just as if that watery material continues to flow into our T-tubule. Let me grab a better pen to illustrate these T-tubules because they're not showing up very well. So here we go. They're just invaginations of our sarcolemma. And then finally, let's not forget that we have our thick and thin filaments inside of our sarcoplasm organized inside of myofibrils. Again, I'm not going to draw that very detailed, but this is going to represent those myofibrils with their thick and thin filaments. So the idea is for all of that calcium that is stored in a high concentration inside of the terminal cisternae is for that calcium to be released and then make it to the thin filaments where our troponin complex is located. Bind with the troponin complex, change its three-dimensional shape, and that will allow for our thick filaments to bind. But how does it all work? Well, from the central nervous system, either the brain or the spinal cord, an electrical signal arises, and we call that an action potential. And that action potential will come down all the way down the axon towards all of the axonal terminals of the axon. And here we see one axonal terminal. And so that action potential, which I'll illustrate with an arrow here, arrives. Well, this action potential consists of the moving around, the flowing around of many ions. It's basically a current. And that is going to disturb the resting membrane potential of our axonal terminal. Every cell in your body is characterized by this gradient of electrical charges across the cell membrane, which we call the resting membrane potential. And only in neurons and in muscle cells, any muscle cells, can that resting membrane potential be disturbed. And when it's disturbed, it results from the opening of various ion channels. So we need to talk ion channels, and we're going to therefore also have to specify ion concentrations. We've already mentioned that the ion concentration of calcium inside the terminal cisternae inside of our muscle cell is very high. Very high compared to what? Compared to the concentration of calcium inside of the sarcoplasm. So I'm trying to write it a bit smaller here to imply that the concentration here is less. But we can't just focus on calcium here. Calcium also plays a role at the level of the axonal terminal. And inside of our axonal terminal, we see that the calcium concentration is lower than outside. So I'm going to write a big calcium symbol right here. I hope you can see that. Let's see if we can make that stand out a little bit better with blue, like so. And so outside any cell, whether it's our neuron or our muscle cell, we're going to see that the calcium concentration is going to be much higher than inside. So our calcium concentration outside our muscle cell is much higher than in its sarcoplasm. But if we compare the concentration of calcium in the terminal cisternae with that of the sarcoplasm, the terminal cisternae is much higher. And remember, ions always follow their concentration gradient. We have to add two more ions. One is potassium. Potassium has, in all of our cells, it doesn't matter which cell, has a much higher concentration inside of cells than on the outside of cells. And the opposite is the case for sodium. So inside of cells, we see a much lower concentration of sodium compared to the outside. And so maybe we can add another sodium here. High concentration there, much smaller here. And the opposite for potassium, much higher on the inside of a cell compared to the outside of a cell. So if there's something that could trigger the opening of ion channels that you find in all of these membranes, then these ions should just follow their concentration gradients, right? If we were to look at the concentration gradient for calcium at the level of the axonal terminal, it would mean that if we could open calcium ion channels in the axonal terminal, that the calcium would flow in. And if we could open calcium ion channels in the terminal cisternae, the calcium would leave the terminal cisternae to go where there's less calcium, in the sarcoplasm. Sodium would flow into our muscle cell, and potassium, on the other hand, would flow out. These concentration gradients that I just showed you, you must memorize because they apply to every cell of the body. And consequently, when you move on to AMP2, you will still have to know these concentration gradients all the way down into your professional field, where these ions play crucial roles, particularly potassium and calcium play absolutely very important roles in maintaining the health of your patients. Okay, so our action potential is arriving from the central nervous system, and this is going to trigger the opening of calcium channels that we find in the membrane of our axonal terminal. And so consequently, in which direction will calcium enter? Calcium will flow, calcium will enter into the axonal terminal. When calcium enters into the axonal terminal, that triggers these synaptic vesicles, which are filled with acetylcholine, to start moving. And when they move towards the membrane of our axonal terminal that faces the synaptic cleft. And they go through the process of exocytosis to then release their neurotransmitters. And consequently, the neurotransmitters start diffusing across the synaptic cleft. So by means of exocytosis, your synaptic vesicles merge with the cell membrane of your synaptic bulb of your axonal terminal, and that releases your neurotransmitters. They will follow their concentration gradient by means of diffusion and ultimately reach the muscle cell. Now, the muscle membrane, the sarcolemma nearby the axonal terminal, is very convoluted. We refer to this part, as a matter of fact, as the motor end plate. That convoluted part is called the motor end plate. And the reason why it is so convoluted is because it is very crowded with protein receptors for your acetylcholine. I will just draw one of these for now, but try to remember that there are many of them. And I'll just draw it like so, and your acetylcholine can bind to it. So this whole motor end plate is packed with these protein receptors. Remember, your cell membranes are phospholipid bilayers with proteins in them as well. And some of those proteins function as receptors, proteins that can bind to something externally or sometimes even internally. In this case, externally, our neurotransmitter called acetylcholine. Well, when this happens, what we see then happening as a result of the binding of acetylcholine is that our receptor changes shape because it's a protein. And when it changes its shape, it opens up to allow for sodium to rush in. So I'm going to grab one of these receptors and I'm going to find a spot here on the board to maybe illustrate better how this all works. So if this is my receptor and acetylcholine has bound to it, that receptor will change shape such that it will create an opening through which ions can move now. And what we're going to see is that our sodium ions will follow their concentration gradients, and our potassium ions will follow their concentration gradients. Notice the concentration gradient for sodium. If we open this channel, sodium will just diffuse in. And at the same time, potassium will diffuse outward. So we see both ions moving through this ion channel, which we call our acetylcholine receptor. And this happens. Some channels are not that picky about which ions pass through them. Now, this is going to impact the resting membrane potential now of our muscle cell. And that is something we need to take a look at next, because that is a start of a new wave of an electrical current that will then spread across our muscle cell down into the T-tubules. And when that wave of electrical current, which we're again going to call an action potential, so when sodium rushes in, primarily it's sodium rushing in and some potassium rushing out, we're seeing a disturbance in the resting membrane potential, which I will expand on in your next video, to where we recreate an action potential. We had an action potential arriving from the central nervous system all the way to the end of our axon here at the bulb. And then we see after acetylcholine binds that that action potential will eventually recreate, and it'll follow wherever there is cell membrane in every direction and ultimately go down these T-tubules. And when this happens, when this action potential moves down our T-tubules, it is going to impact the ion channels in our terminal cisternae, more specifically calcium ion channels. So the arrival of the action potential down in the T-tubules is such that it's now in close proximity to the calcium ion channels in our terminal cisternae, such that calcium will be released. And this is again passive transport, diffusion. Our calcium ions are just following their concentration gradients. And eventually some of that calcium will collide with the troponin complexes of our thin filaments, and you know what happens next. Our thin filaments will then start undergoing cross-bridging with the heads of the thick filaments, such that the heads can pull the thin filaments into the H zone, causing contraction. I think we'll stop here, and we're going to spend some more time discussing resting membrane potential and what it means when it is disturbed as a result of the binding of acetylcholine, such that we see our sodium and potassium moving. Till next time.